Okay, so one of the approvals has been the Tisagenlecleucel, which was actually approved for pediatric young adult ALL. What do we know about that data?
So this data has really been impressive and transformative. In this data, patients who were either relapsed after transplant or multiply relapsed or primarily refractory, 82% of them went into MRD negative remissions, which is incredibly impressive and led to the approval. But the even more impressive data is that at ASH this year we have seen a follow-up abstract that has shown durability of response at 18 months to be 67% in patients who achieved that remission. So this has really transformed the pediatric young adult ALL field, where CAR T cells are not only very, very good at taking highly refractory patients and putting them into deep remissions, but some of them appear to have long-term remission and potentially cure from the CAR T cell product by itself.
We have other options for pediatric young adult ALL. Is this really an advance?
It's an advance because previously these particular patients, the patients again that qualified for this trial, had to be multiply relapsed or refractory. And transplantation for a patient who has refractory ALL just simply doesn't work. The survival is well less than 10%. Even minimal residual disease prior to transplant means very poor outcome in a patient with B-cell ALL. So this is really truly transformed the field, taking patients who we just used to send to hospice and giving them a viable opportunity for cure.
So if I remember right, that approval goes up to young adults up to through the age of 25.
Correct.
If you're an adult, then...
If you're an adult, what do you do?
Right.
So this is very important. There are a lot of data now, data that was presented last year at ASH and this year at ASH in the adult population, and there's now a couple of significant papers in the adult population showing that with both CD28-based CARs and 4-1BB-based CARs, that patients can achieve similar rates of remission, but the big caveat is that toxicity has been noted at what appears to be higher rates in the early phase trial. Some of the trials have had to have adjustments made such that doses, one trial split doses and found a better toxicity profile. Other trials have had to cap doses at certain amounts in order to get better toxicity. So clearly, the efficacy when the dose is correct has been good. It's been in the 80 to 90% remission rates. The big challenge, though, that we have in the adult population is toxicity. And as that is solved, I think that this should be able to be moved more into the adult population.
I've been impressed with some of the early studies in neurologic toxicity in the ALL trials, at least some of the JCAR study, the JCAR 15, I think. Can you talk a little bit about that, or what?
The big problem, of course, early on was the severe fatal neurologic toxicity that occurred in a small number of patients. And this has been seen across in a number of different trials. Diffuse cerebral edema occurring, and it's just catastrophic.
We know the mechanism of that?
The mechanism is unsure, but it's probably similar to the mechanism of other neurotoxicities, but likely a very severe degree of that particular toxicity. There's been some nice studies that have shown that CAR T cells lead to significant permeability in the CNS, and very beautiful studies out of Seattle that have shown that this permeability leads to a marked increase in a wide variety of cytokines and in cells getting into the CNS. And that's likely what causes neurologic toxicity of all different types. But it's also a benefit because you want those cells in there because they're very good at treating CNS leukemia. So we need to find a good balance with that.